Dear colleagues, and the next presentation would be made by Dr. Sычев, who is the lead of clinical pharmacology chair and director of Russian Medical Academy of Continuous Post-Diploma Education. Professor and correspondent member of the Russian Academy of Sciences. And the topic of our presentation today is the technologies of personalized medicine, the view of clinical pharmacologists. And we all know, so when we speak about our RWD and RWE, especially about most innovative drugs, orphan drugs, then immediately we face personalized medicine as essential parts of that. Where we can use real-world data and real-world evidence, RWD, RWE. Good afternoon, dear colleagues. I'm welcoming all participants of this really unique conference on the real-world data and real-world evidence. This is very symbolic event for our medicine, for clinical pharmacology, this type of the conference, as soon as the publishing of this unique monography. My presentation is devoted to the technologies of personalized medicine from the point of clinical pharmacologists, and I will show how real-world data related to that. We know very well that it is academic Worchel many years ago in 1961, in his very famous monography, he wrote that everything should be administered 3 times a day, except sleeping pills and anti-diarrhea pills. So now we have so many drugs, and in the essential drug list, we have about 800 drugs with international non-patented name. We have methodology of EBM, evidence-based medicine. We have clinical guidelines based upon the principles of EBM, and there are the results of big randomized clinical trials, meta-analysis, systematic reviews, etc. So we have everything available in order to treat patients with drugs effectively and safely. But still, it doesn't go very smoothly because Roszdravnadzor registers about 30,000 adverse events, non-favorable, reported by the doctors. And but if you use this method, like local trainer to and monitor adverse events in the medical hospitals, in the medical organizations, so it can be 10 times more. So you can, in reality, see more than 300,000 adverse events a year. And if you look at the structure of adverse events, which is shown here, for example, bleeds with anticoagulants, we see that, in fact, 1/4 of the patients who had bleeds with anticoagulant treatment, doctors violated something, either the label or clinical guidelines. In 2/3 of the cases, specifically, doctors administered the drugs correctly without violations of the label, but still adverse events may happen because they depend on the patient factors. Individual pharmacological response can be guided by the gender, by age, by genetic factors, drug-drug interactions, comorbidities, and sometimes lifestyle and genetic factors as well. So all that in the basis of search of particular biomarkers that could tell us what are individual special features of the patient in order to predict the effect of the drug response or non-response and to have very personalized approach. This understanding of the need of personalization, especially personalization of drug treatment, is reflected in the strategy of scientific and technological development of Russia, where it's mentioned that the biggest priority is the switch to the personalized medicine, and specifically due to the very rational use of drugs. But in this context, we speak a lot about 3P, 4P, 5P, 10P medicine, but the issue of personalization of pharmacotherapy is reflected in one more strategic document, which is strategy of healthcare development up to the period of 2025, where it's mentioned that personalized pharmacotherapy, in particular based on biomarkers, is our priority for the healthcare development for the next 5 years. In 2018, the ministry issues this document. This is concept of 3P medicine, and personalized medicine is the individual management of the patients in terms of measuring of indicators, physiological or pathological, that characterize patients' portraits. So these indicators can be measured, and they are called biomarkers. From the point of clinical pharmacology, definitely we want to have biomarkers that indicate how the drug is working in the human organism, what is the doses, what is the distribution in between the tissues, and definitely we would see the markers of pharmacodynamic sensitivity, specificity, etc. We want these biomarkers be predictive, specifically when the patient is still in the therapeutical window of particular concentrations. And these are exactly biomarkers that are reflecting the work of the transport system and enzymatic systems. So biotransformation of phase 1, 2, so-called dreams that individualization is possible due to the prediction of pharmacokinetics also refers to our teachers. And this is Professor Larkin, who wrote back in 1981 about the technology of personalized medicine. He wrote that we would be closer to personalized treatment when we know how to manage the effect of the drug in the patient's organism. You know that partially we already start doing that, and we have some tools that help us to control the effect of the drug in the patient's organism. So these biomarkers mainly focused on the fact that we could help our patients to select correct patient, to set correct diagnosis, to select correct treatment. So the drug should be administered on the right time, on the right dose, to the correct patients with good response, with absence of adverse events, and good effective outcomes and very hard endpoints like blood pressure, like response to treatment, etc. So this classical chart distribution of our patients in the speed of biotransformation, this is specifically shown here. We see that the population is very homogeneous, non-homogeneous, and you see that some patients can metabolize beta blockers faster. There are ultra rapid metabolizers, so there are extensive metabolizers, intermediate and poor metabolizers. Mainly that is guided by the genetics, and these are minor changes in the gene where one nucleotide is replaced by the other nucleotide, and it changes the gene expression, and we see that conformation is changed and activity is changed, and finally the biotransformation speed is changed. It can be either slowed down or facilitated. And this is exactly the basis for the understanding of the need of personalized, specifically dosing of drugs. And here are the methods of clinical pharmacology. Personalization is entering this field. These are markers of personalizations. The most clear is pharmacogenetics, pharmacogenomics. Then we use PCR and sequencing, specifically new generation sequencing. Also big hopes on pharmacotranscriptomics and these biomarkers, microRNA, and especially plasma microRNA, and the system here is used. This is the system of transport and biotransformation of drugs, and pharmacometabolomics, which is HPLC and phenotyping, CYP, and so transports using again the activity of metabolites and phenotyping of cytochrome P450 typing and different enzymes of biotransformation and transport system. And main method here is HPLC with mass spectrometry detection. Well, PCI is getting cheaper from year to year, but HPLC is not getting cheaper, unfortunately, and machinery is improving, but still the cost price is pretty high. Pharmacogenetics first was mentioned long ago. First book with the title Clinical Pharmacogenetics was published back in 1978. And the main author here is Professor Skapuna from Ternopil, from Ukraine. And this is the letter to WHO. It was dated back in 1975. And here we understand that this is very perspective in the aspects of pharmacotherapy individualization. In our academy, the issues of personalization we are dealing with them for several years already, and this is very serious at the level of our Department of Molecular Medicine and Personalized Medicine. And here on the slide, you can appreciate the spectrum of drugs where these biomarkers, personalized biomarkers, can be studied. And here are drugs of psychotropic drugs, anticoagulants, oncology, ophthalmology, painkillers. So you see, these are big areas, and here are lots of departments. And you see that multiple stakeholders are involved. So the design of these biomarkers is at a very different stage, but nevertheless, it's actively ongoing in the state order and also grants of the Russian Foundation for Fundamental Studies. So we came to the conclusion that the study of these biomarkers should be systemic. And let me show you the real world data that we received here in this cascade of studies. The first thing we need to do is to understand what sort of challenge we are going to resolve in terms of personalization. We want to reduce the adverse event rates, increase safety. So for that, we have very good tools that we clinical pharmacologists know that. We know how to monitor, we know the principles of pharmacovigilance, and then we understand the real picture on how to use the drugs properly, on the one hand. On the other hand, we identify those patients who had, for example, some adverse events. Then we do the biopool, then we identify DNA markers, and then we select particular molecular or genetic biomarker, and then we're checking, and we can refer it to the group of non-adequate response with adverse events and to the group without adverse events. And here is a very big place for the real world studies, because this is exactly what we need. And based upon these studies using bioinformatics, we are designing models, algorithms of personalization, and later on we try to validate them and then randomize them. And then in non-randomized studies, it also refers to the real world studies. And based upon that, we are trying to formulate evidence-based data that this personalized approach is working. It's available from the point of efficacy and safety, economical analysis from the point of health economy, which is very important, as the studies are small and not supported by large companies, mainly for the grants. We want to increase the level of evidence using meta-analysis. So meta-analysis, this is what we do in order to provide this data and implement these approaches in the clinical guidelines, in the standards, and through the educational tools. Here on the slide, here you see where there is a very big place of the real world data. So to do high quality studies, large randomized clinical trials, normally it's very costly, very expensive, and the experience of other countries shows that sometimes these studies are terminated because of lack of financing. That is why the good level of evidence with good level of data is not always possible. Let me give you the examples of such trials on the methodology I mentioned before. So the first study, and by the way, I am presenting the results, and these results are published in the journals with very good impact factor. So you see, this is one of the publications where we studied the spectrum of adverse events. These are neuroleptics in adolescents with first acute psychotic episode. And we showed that in a number of adolescents, antipsychotics that they are treated lead to the adverse events. So biobank was formed and the registry of these patients as the basis for the future associative studies. And then studies that show the association between the challenge and the biomarker that we have selected. So biomarker could be genetical. So that would be principal gene candidate approach. So that's classical approach. So, for example, metabolizing of cytochrome P450, then it acts vitamin K and the cycle of vitamin K, so pharmacodynamic marker, and we study polymorphisms in these genes. And the experience shows that the studies of these candidate genes polymorphism can identify the sensitivity to warfarin. So this is how we know that warfarin is metabolized in liver with the effect of cytochrome P2C9 and then influences the cycle of vitamin K and inhibition of VK or CL1 in the liver. So this is our data. Well, sometimes, especially for the drugs that are used for a long time and that are already designed in Russia, it's very difficult to select the biomarker. And then we are supported by the methods of bioinformatics and in vitroma methods. And for example, phenazepam, which is actively used for more than 60 years. So currently in Russia, this is most frequently used benzodiazepine in the Russian Federation. So in order to predict a particular adverse reaction and to select the biomarker, then we need to know which sort of cytochromes or isoenzymes it is metabolized. So we referred to our colleagues from the Institution of Biomedical Studies, and with their support, using in silico approach and in vitro modeling, we could predict cytochromes that could metabolize this molecule. Then, using special bioreactor, and that is Professor Talevitsky design. So in vitro, we made the model and it showed that these predicted in silico cytochromes in vitro in human hepatocytes metabolized phenazepam. When we studied polymorphisms of cytochrome P450, we showed that it is them that can predict very serious adverse events, phenazepam, and this is how we can have personalized approach, and we see that this is not expensive. So possibly this approach would be working. So this is a second quartile study. This is drug metabolism and personal therapy. So we published this data and they liked methodology of these studies, the search of candidate genes. And this is a new topic we cannot avoid, which is COVID-19. So here are searches of biomarkers of severity. And by the way, there are searches of biomarkers of efficacy of those drugs that we are trying to use in the COVID-19 as pathogenetic treatment. So they say that, for example, many drugs that are positioned that ready-made products like hydroxychloroquine. So the failure could be explained, but it worked for some patients, but for some it didn't. So we need to find simply biomarkers of efficacy and safety of this specific example, hydroxychloroquine. And this is definitely not our study. This is the study of genome-wide association of severe COVID-19 with respiratory failure. So this is very recent publication where investigators performed this genomic association analysis in different severity. So this is a full genomic analysis. They identified several molecular and genetic biomarkers that were associated with more severe course of COVID-19 that required invasive respiratory support in these patients. And by the way, I can confirm that currently we are completing the publication where we study in our Russian ethnic groups, very different ethnic groups, we have very good collection, the frequency of the same polymorphisms that were shown in this publication, association with the severity of COVID-19. So, and we are trying to refer that to the epidemiological data. So, and it appears to be a very interesting picture. So that will be our joint publication with the group of Professor Polomursky from the Institution of Vavilov, Institution of Genetics, and I will share this publication as soon as it is ready. So that is very promising. So genomic-wide associative analysis, GVAS, that's new method for the search of new molecular genetic partners in the personalized treatment. And specifically, it is used for quite old drugs, definitely in new situations. So this approach, GVAS, was used as a method to search candidate genes biomarkers of bleeds with marine anticoagulants. And so there were some genes that we actually knew them, but we confirmed our data in this genome-wide association study. And all that confirms the real resultativeness of this study. So here are a few our results where we use the same approach. This is real world studies. And this is a publication on the association of cytochrome P dissociation and omeprazole, hydroxyomeprazole in the urine. So that speaks for biotransformation rate of biometrazole in patients. So this is urine metabolic ratio of omeprazole in relation to cytochrome P2C19. Really slowed down in metabolic phase. And so we could expect, let's say, more prolonged and pronounced effect of the drug. So after this GVAS, genome-wide association study between the efficacy of omeprazole, which is proton pump inhibitor, um, uh, and we analyzed it in relation to peptic ulcers, we finally published a meta-analysis shown here. We included our study here, other Russian studies. So using this meta-analysis, small studies in the real practice, we showed that truly the carriage of second allele variant associated with a more effective anti-Helicobacter eradication therapy in this category of patients for the personalization. And this is exactly what we do. Well, in the conditions of real practice, it's not enough to find association between the challenges of drug treatment and particular biomarkers. Based upon these studies, we can create some models, algorithms to make clear understanding how to select drug based upon genotyping. With that, we already know several algorithms. So from very simple dichotomic, for example, algorithm of Tigger-Lor. This is American algorithm, American consortium of pharmacogenomics based upon cytochrome P polymorphism up to very complicated algorithm regression model. This is warfarin dose prediction. Or for example, very special scales. For example, this is a scale of resistance to clopidogrel based upon genetic and non-genetic factors up to simple information, food for thought for the doctor, that there are particular genetic problem of the patient and you can select proper drug. So this is Canadian Diabetological Society. So this is how they select in atrial fibrillation old coumarins and NOACs based upon different factors, specifically including genetic factor. Well, this is all food for thought, additional argument to make personalized decision. It's important that these algorithms have been designed with the help of our friends bioinformatics. And this is a very successful cooperation with the High School of Economics, and this is Faculty of Computer Science, where we try to create a model of prediction the outcomes of the mortalities of the patients with atrial fibrillation after the coronary artery stenting who get dual or triple antithrombotic treatment, so clopidogrel, aspirin, and anticoagulant. With that, we took into consideration genetic, pharmacogenetic, and then using artificial intelligence, our colleagues created 3 models. And this one was predictive with probability of 100%. Next 6 months, the probability of mortality of the patient. So in the model here, we had genetic and non-genetic factors and also modified factors. And with the help of modification, the doctor could change modifiable factors and finally change the prediction, the forecast, the fate of this patient. So very hard outcomes. Again, this is very valuable approach to personalization, which should be checked in the studies. It's important that these biomarkers, specifically those we showed before, very differently spread in the Russian Federation. The topic of pharmacogenetics was always interesting to my teachers. So this is Professor Suleimanov, these are several early publications, but now we study, it's in a large scale, we see polymorphism variants in different ethnic groups of the Russian Federation. So definitely it should be all taken into consideration. And now geography is very large and many clinical pharmacologists participate in this project. So we showed that, for example, there are differences in polymorphism of genes that guide sensitivity to warfarin in different Northern Caucasian populations of Russia, or the carboxyesterase 1 gene polymorphism associated with a low risk of bleeds with the bigger drum. It's also different between Russians and Northern Caucasian republics. So there should be some regional approaches depending on the ethnic composition of the population. This is also option to personalize. Definitely, you never reach true personalization without cost efficiency. So this is health economy analysis, comparative clinical and economic evaluation of pharmacogenetic testing implication for the bigatron in patients with atrial fibrillation. So this is Dr. Gerasimova publication. And this is another analysis on genotyping on carboxyesterase 1 to predict lower risks of bleed with the dabigatran in patients with atrial fibrillation when you select the dose between 110 and 150. And it was shown that if you use this method and correspondingly, you can save the resources, which is economically feasible. Except that in order to implement it into practice, we need to definitely give the system of decision making support, where the doctor is using patient data and then clinical data from the lab, and then finally the software could provide the, let's say, prompt for decision making. This is like Dr. Tsvetov, PharmaSuite, and also you see PharmSuite Pro, and also another system for predicting adverse events on psychotropic treatments based upon genotyping on cytochrome P450. And this last system in decision making, this is real world data. It was tested and published in human psychopharmacology. It was shown that this personalized clinical decision support system for antipsychotics in patients with anxiety disorders based on the pharmacogenomic markers. It's increased safety and efficacy. So this personalization really works. Definitely, our data, we try to make it cumulatively, not only our data, but also the data of other investigators from the Russian Federation. And last year, we managed to publish in Pharmacogenomics. So this is one of the leading journals in the pharmacology world, this systematic review on the future outlook for Russia, speaking about ADME pharmacogenetics. So we are very proud by that publication, as those who added some evidence-based data due to the Russian impact. So wrapping up, I would like to say that implementation from the point of technical pharmacologists from the evidence-based data should be complex. And here the study of the real practice, here real-world studies take the central position for the collection of the evidence, for the assessment of the technologies and implementation through the clinical guidelines and the standards of treatment in the clinical practice. So wrapping up, I would like to quote one of our teachers. The dream of our teachers about personalization are closer to the reality, and academic culture. I already started with him. Now I'm finishing. The scheme for the doctor is good in good cases when there is no time to think or no capability to think, and definitely no offense meant. That's why there is a continuation. There is nothing to think with, not because of cognitive dysfunction, but no food for thought. So these technologies give the doctor the chance how to make individual decisions, personalized decisions for the patients based upon the study's results. My final slide is here. Methods of personalized medicine should not be contradicted to the evidence-based medicine. So they should work together, because these are the same tools of evidence that are used for using evidence and personalized methods to use it in the real clinical practice. Thank you very much for your attention. I do apologize. I cannot participate in this event offline. So if you have questions, I'm ready to give answers in the electronic mail. Thank you very much. Many thanks to the organizers, and personally to Alexei Kolbin, my congratulations. Many thanks to the association. I wish you a fruitful day. Thank you very much.